Welcome to The Explainer. Today, we're going to pull back the curtain on something that happens in hospitals every single day, but is way more complex than you might think: a blood transfusion.
So what is it? Well, it's not just one thing. It's actually this really intricate, multi-layered safety system. And once you understand how it works, you'll really appreciate the amazing science that's keeping people safe every single day.
Yeah, this is the absolute worst-case scenario. It's called an immune-mediated hemolytic transfusion reaction, and it's basically when your own body's defense system sees the new blood as a threat, like a foreign invader, and just goes on a full-scale attack. It's incredibly dangerous and can even be fatal. So the big question is, how do we stop that from happening? Well, it all starts way before that bag of blood even gets near you. It begins with a whole process called pre-transfusion testing. You can really think of this as the entire security setup. It's got all these different checkpoints, some are just paperwork and procedure, others are hardcore science, and they all have to work together perfectly. So let's get into the nitty-gritty, the lab work itself. Okay, this right here, this is the heart of the whole operation: compatibility testing. This is where the lab techs become detectives, analyzing both blood samples, looking for any sign of a potential clash. And a really key piece of this puzzle is what you could call the final dress rehearsal. And that's the cross-match. I mean, it is literally the final check. You take a little bit of the patient's blood, mix it with a little bit from the donor bag, and see what happens. You're basically asking them, hey, can you two play nice together right there in the test tube? Now, here's the thing. This entire beautiful chain of safety, all the fancy science we're about to talk about, it's all completely useless if we don't get the very, very first step right, and that is getting a perfect blood sample from the patient. And believe me, the rules for that sample are absolutely non-negotiable. We're talking 2 different patient identifiers, a fresh collection date, super clear permanent labeling. Every last detail is checked and double-checked. Why? Because one tiny mistake here at the very start can completely torpedo all the science that comes after it. So let's see how this all plays out in the lab. It's basically a 5-step dance. First, you test the patient's blood, ABO, RH, the whole deal. Then you double-check the donor blood to make sure it is what it says it is. After you've picked a unit, you do that cross-match we just talked about. And for the final layer of safety, you check everything against the patient's past records. See? It's not just one test, it's this whole series of checks and rechecks. Okay, let's zoom in on one really important part of that patient testing, the antibody screen. Think of it like a background check, but for your immune system. We're trying to find out if you've ever made any unexpected antibodies, maybe from a past transfusion or a pregnancy, that are just kind of lurking in your system, waiting to cause trouble. Now, here's the key thing to understand. We're not looking for every antibody, just the ones that could actually be dangerous. And how do we know if they're dangerous? Well, we call them clinically significant if they cause a reaction at normal body temperature, 37 degrees Celsius. We even have this special stuff, anti-human globulin, or AHG, that helps us find the really sneaky antibodies. If we see a reaction under those conditions, that's a red flag. That's a potential threat. So all of this work, all these tests, they're all leading up to this one final checkpoint, the cross-match. This is the moment of truth, where the donor cells and the patient's serum finally meet face-to-face in the lab, right before the real thing. And what's interesting is that there are different types of cross-matches. On one hand, you've got the immediate spin. It's fast, it's simple, and it's basically just a final confirmation of the ABO blood type. We use that when we're pretty confident everything's clear. But if that antibody screen we talked about turned up something, or if the patient has a history, then we have to pull out the big guns, the anti-globulin cross-match. It's a much more involved test designed to catch those trickier, more subtle problems. Of course, this is the 21st century, right? So now, a lot of the time, we can actually swap out that simple physical test for something even faster, the computer cross-match. But hold on, this isn't as simple as just typing names into a computer. The safety rules are, if anything, even stricter. The computer has to have at least 2 previous matching blood types on file for that patient. The patient's current antibody screen has to be clean, and they have to have 0 history of any significant antibodies. If all those boxes are checked, and only then can the computer give the green light. It's a really cool example of how we can use data to make things even safer. Okay, so this whole system is great. It's thorough, it's safe, but it takes time. So what happens in an emergency?
A major car accident, a patient is bleeding out, and you don't have hours, you have seconds.
Well, the system has a built-in break glass in case of emergency button. That's where Group O, Rh negative blood comes in. It's the universal donor. That means pretty much anyone can receive it safely. It is the ultimate fail-safe for those life or death moments when there's just no time to run the tests. The priority is to save the life, period. So we've gone through all this high-tech lab tests, smart computer algorithms, all these complicated procedures. So after all that, what do you think is the biggest risk? Where does the system most often break down? The answer might surprise you. It's not the science, it's us. The number one cause of fatal transfusion reactions is simple clerical error. Mislabeling a sample, grabbing the wrong bag of blood from the fridge, misidentifying the patient at the bedside. And that is why those super strict rules about labeling the sample tube at the very beginning are, without a doubt, the single most important safety step in the entire process. Now, the good news is that the technology inside the lab is always getting better to help reduce those chances of error. You've got things like gel testing, which uses a gel that literally acts like a sieve, making the results way easier and clearer to read. Then there's solid phase testing, which is super sensitive and great at picking up on really weak antibody reactions. All these new tools are constantly making the lab work more reliable, more accurate, and more standardized. And that really brings us to the final big idea here. As the technology gets smarter and handles more of the complex science automatically, what does that mean for us? It means our focus has to shift to perfecting the human parts of the job that technology can't replace. The future of transfusion safety isn't just about finding a better machine. It's about perfecting that crucial partnership between amazing technology and a careful, focused human being.